Hello.
Hi.
Shelter in place in Cambridge.
Mm.
Once we start, I do want to be able to see you. And we'll start in, we'll give you one extra minute, so we'll start at 12:39. While we're waiting, does anyone have any questions about the project? I'm still waiting. Most of you haven't answered yet as to whether you're going to be doing the reenactment of the murder or whether you're going to talk about the hearing. Is anyone who's present here going to be doing the reenactment of the murder? Is anyone going to try that? No.
Yeah, my group is.
What?
I think my group is doing a reenactment.
Okay. One of the consequences of teaching this class is I've become very disheartened with the criminal justice system. It's just there's so much inequity. It's and it's so endemic, right? It seems so chronic. It's really upsetting. Okay, I think we'll get started. Again, I really would like to see all of you, and so I'll warn you. Unless you email me and explain to me why your camera is broken, I would like to be able to see you. I think it's more better, and it makes us a bit more accountable in some ways, I think. Okay. I'm going to start off with where we left off, and I said, Amar, are you here right now? Amar? Oh, wait, you know what? Before I do this. No? No, Amar?
I'm here.
You are here. So you were having trouble with, if I remember correctly, um, you wanted to look at I want to go over the precipitin test again before we move on. So
Okay.
no, that's not what I meant to do. So let me just go over that again. Let's try that again. Why is it not coming up? I don't understand. Embarrassing. What is this? No. No. Give me a second. Sorry. Give me a second. This one I have to do. Turn that on. Close that. Save. Okay. Now. Okay. Now. Good. Now it should work. Okay. Okay. Sorry about that. Okay, so this is the precipitin test, and we were using this to figure out blood, with the source of the blood. Is it animal or is it human? And so let me just go back to the principle here. And the principle here is this. This is the precipitin test. And what's happening here? So what you do is, in a test tube, it's a slightly different format in the next slide, but it's the same principle. You put a solution. Can you see the arrow here, guys? Can you see this? Can you see the arrows?
Yeah.
Can anyone just?
Yeah, we can see it.
Yes?
Yes.
Mhm.
Thank you. Okay. So okay. So you put antibody in here, and then you put a solution of antigen on top of it. You carefully layer it on top. So you have two solutions here, one antigen and one antibody. And at the interface where the two are meeting each other, they start to interact. And what happens is that the antibody binds the antigen, and it forms these large clusters that we talked about, because antibodies have two ends. It's a fork, right? And so what happens is you form these large clusters, and it precipitates, and it forms a solid. And you see that here. You form this solid here. Okay. Amar, does that make sense?
Yeah.
Okay, so now what we're doing in this slide here is we're simply, let me move this over here again. What we're doing in this slide here is we're doing the same test, but in a gel format. This is like a gel where you would run DNA. It's gelatinous, made of agar, and you make a series of little wells in it. You're looking down at it, okay? You make a series of wells, and into each of these wells, you place some antibody that's from against a different animal, human, dog, cat, fish, etc. Right? And then in the middle, you place the blood from the crime scene, right? And then what happens is that the substances in the blood slowly diffuse in all directions from each of the wells, and you get interaction between them. And if the antibody, so in this case, the antibody in the human is binding to some antigen that's in the crime scene, blood, right? And it's forming this little white precipitate at the interface between the two. And so that's how we know it's human. It's just like what's happening in the test tube here, where you get this precipitate forming between the two, you get this precipitate that's forming between the two. Okay, so that's all that is. Any questions on that? So classifying blood. And I was mentioning before, there's over 250 different blood types that exist, and these are based on different proteins that are on the surface of the red blood cells. Medical people and forensic scientists are not interested in any of them except two, and those two are the ABO system and the Rh system, because those are the only ones that produce strong immunological reactions that antibodies bind against or bind to. Okay, so now the U.S. distribution. This was the question of just to throw that in there of the blood types. There are 4 blood types: A, B, and O, and O is the most prevalent, followed by A, B, and lastly by AB. Okay, now the principle is, the question is, what is in your cells versus what is in your plasma? So if you are blood type A, what does that mean? That means that you have, what is the antigen in your blood? Which of the glycoproteins do you have on the surface of your red blood cells? A, B, you have either have A, you have B, you have A and B, or you have none. That's what this means. So if you're blood type A, it means that on the surface of your cell, you have this type A glycoprotein. It's a specific protein. People don't even know what function it serves. This person has a different version of it. It's the B glycoprotein. This person has both A and B. And this person with O blood, you don't have any of the either A or B glycoproteins in your blood. So then the question is, what do you have in your serum? And the principle is that you have in your serum whatever you don't have, whatever blood type, against whatever blood type you don't have. Okay? So for example, if you're type A blood, are you going to have any antibodies against the A glycoprotein? What do you think?
No.
No.
No, right? Because what would happen if in your serum you had an antibody against the type A glycoprotein? What process did we talk about yesterday would happen? What kind of things would go wrong? What would happen?
Macrophages would eat it.
That's right. It would clump up and the macrophages would eat it. So what antibodies do you have in your serum against what glycoprotein?
B.
B. You have an anti-B. I'm just going to put B, but you should write anti-B, just so we're not being, I'll write it. I'll just put it for this first one. It's just, it's so hard to write in this thing. Anti-B antibody. Okay? So therefore, what antibody are you going to have in your serum if you're type B?
Anti-A.
You're
Anti-A.
going to have anti-A. What are you going to have if you're AB?
None.
None. Bing.
None. And what are you going to have if you're O?
A and
Anti-A.
Right, you're
Anti-A
gonna have
and B.
antibodies against A, and you're going to have antibodies also against B. So that's the principle. So that tells you what you have in your blood and what you have in your serum or your plasma. Okay. Can I erase this?
Dr. Hornstein,
What?
what's the difference between the serum and the plasma again?
Someone else tell me, guys. Someone, you can say it out loud. What's the difference between serum and plasma?
Serum doesn't have the clot thing.
Say it louder.
Serum doesn't have the clotting part.
That's right. Serum is missing the clotting factors. The way you make serum is to induce clotting. So you get rid of the clotting factors.
I see. Okay, thank you.
You're welcome. Okay. So that's just said that. We can go. Okay, now let's just talk about transfusions for a second, because a patient cannot receive an antigen to which they have an antibody or a major transfusion reaction will result. So again, what I want you now to do is, let's do the first one together. You tell me, what is the antibody I have in my plasma if I'm blood type A? Anti what?
B.
B. Okay. So, all right. Sorry. Anti B. So therefore, I can receive a blood transfusion from what person with what blood type?
A
Now,
or
I just
O.
want to make something really clear here. When you give blood, they separate out the plasma and the blood cells. So when we say you're getting blood, we're talking about the blood cells themselves. We're not talking about the serum, okay? So if that's the case, if you receive a transfusion of blood, what kind of blood can you get?
A or
B
O?
and O.
A and O. Why can you not eat? Why can you, I understand B, why can you not get blood type AB? Someone tell me why you can't get blood type AB.
AB has both A and B, and since you're A, you don't have the antigens for B, so it will just coagulate.
Good. Okay, so this one is what? So what can you receive here?
For B, you can receive B and O.
B and O, right? And AB?
A, B, and
A, B,
AB.
and O.
A. Okay, so so let's think of it another way. You have, what antibodies do you have here? You have none, right?
None.
So you can receive everything. You can receive A, B, A, B. All, I'm just gonna wait. I'm just gonna write all. Okay. And what about O? What is he
O.
going to be able to receive?
O.
O.
Only receive O, right? Because he has antibodies against all of those A and B. Okay, good. So the last thing I want you to do, and this one I'm going to have you to do, try to do it on your own. And we call front typing and back typing. So if you have access to blood, if you have a crime scene, you only have access to the blood cells for some reason, which is unlikely, but let's just pretend. You do what you'd call front typing, whereas you add antibody and you look to see whether it agglutinates. So you would add A, anti-A, and then you anti-B, and you try to figure it out. But you can also back type. If you just have access to serum, then all you do is you add cells. You add either A cells or B cells, and you look to see whether you get agglutination. So that being the case, oh, didn't I have a chart for you to do there? Did I leave that out? Oh, there it is. Okay, good. So I want you to fill out this box. So wherever there's agglutination, you put an X. So again, this is front typing where you're adding, your sample is blood cells and you're adding antibody. And this is back typing where your sample is serum and you're adding cells to type the serum. Okay, so go ahead and fill these boxes out together. I mean, not together, you can't do that. But go ahead and fill them out. Again, wherever you see agglutination, I want you to put an X. Okay, I think just 10 more seconds. Okay, let's try it. So let's go down the row here first. If you're type A blood, and you add type A blood cells, and you add anti-A body, you're going to get agglutination if you add anti-A antibody? Anybody? Are you going to get agglutination here? Are you going to get agglutination here? Yes or no?
Yes.
Yes. Okay, good. I'll make a check mark. Are you going to get, if you're type B blood, are you going to get agglutination?
No.
No. What if you're type AB?
Yes.
Yes. And type O?
No.
No. Thank you. Whoever, I couldn't see who was talking, but I appreciate it. Okay, now you're type anti-B antibody. What, you got a check mark here?
No.
No.
No. Check mark here?
Yeah.
Yes.
Yes. Check mark here?
Yeah.
Yes.
Yep.
Check mark here?
No.
No. Good. Okay, now here you have the serum. You have the serum of someone who's type A, and you add A cells. No. Are you gonna get, sorry, I was looking at something else. If you're type A blood and you have the serum from someone who's type A blood and you add A cells, are you gonna get agglutination?
No.
No.
No? Well, let's think about it. The serum, can't do that. The serum, if someone's type A blood, they have the anti-B antibodies, right?
Mm-hmm.
So are they going to get agglutination if you add this anti-B antibodies to type A blood? Are you going to get agglutination? No.
Sure.
Right? So none. So now here's the serum from someone who's type B blood, which means they have the antibody against A, right? If you have the antibody against A, are you going to get agglutination here? Wait,
Yes.
what am I doing wrong? If you're type A blood, you have anti-B antibody. So if you add anti-A antibody to that, you have anti-B. Wait, let me stop again. You have anti-B antibodies here, right? And you have A protein. So therefore, you are not going to get agglutination. Here, you have anti-B antibodies and you have anti-B cells, so you are going to get agglutination. And because you have B on the cells, you are going to get agglutination. And you are not going to get agglutination here. Here, if you have B cells and you have type A cell, if you're adding B cells to type A serum, it's anti-B antibody, you're going to get agglutination here, not here, but here, like that. Does that make sense? Does anyone have any questions? Okay, sorry about that. Okay, pretty sure that's right. Sometimes I get a little confused, but I think that's right. Okay, I'm going to move on. Okay. Okay, the RH system. The RH system is just what's called the D protein. Instead of the A protein or the B protein, it's the D protein. So you are if you have it, you're RH plus. If you don't have the D protein, you're RH minus. And it's also, the D protein is also known as RH antigen. They're synonymous. So this person, for example, would not simply be AB. This person would be AB plus. And if this person did not have the RH antigen, they'd be AB minus. And if they had the RH antigen and neither of these, they'd be, what would it be if this person had neither A nor B, but they did have the RH antigen? What would be their blood type? Go ahead, just type it in. What's the blood type of someone who has neither A nor B antigen, but they have the D antigen? Yeah. Not just O, guys, it's O positive, right? O positive. Good. Okay, good. Now, the last thing you want to do is can you originate the specific, so now we've narrowed it down. So if you find blood and you blood type it, you narrowed it down to a class of people who have that particular blood type. It can be very useful, right? If none of the suspects have that blood type, then you can pretty much guess that you can let them all go. So sometimes knowing the blood type is going to be sufficient to move yourself along on a case. But we want to try to find the exact person. And to do that, what component, I think we answered this before, what exact thing do we need to know to find, what part of the blood do we want to find the person exactly?
DNA.
Sorry again? The DNA, and that is in which type of cell?
White blood cells.
White blood cells, thank you, good. Okay, good. Okay, so you guys read this last night, I hope, and again, this was a huge, as you might have guessed, this became a thing in the 80s and 90s. And it's a really tragic case. And I just want to look to see, it's 1 o'clock. We have 20 minutes. So I wanted to do a soft test of the, stop share for a second. I want to do a soft test of the breakout rooms because I want to start using breakout rooms. And I was reading about it last night. So let me see if I can do this. And then I want to have you discuss a couple questions once you're in the breakout room. So let me just see if I can do this. Give me a second. You can talk amongst yourselves quietly. I want to just try to do this. So here we go. So I am going to, how many of there are 48. And if I want 4 people in a room, that's 12 rooms. And I want to create the rooms automatically. Okay, so here we have all the rooms. So what I want you to do, here's the rooms. You can't see this. Can you guys see this? No, you can't see
No.
this. Okay, so I've broken you out randomly into rooms. So let me go back now that I've done that. Okay, and I want to go here, and I basically want, okay, there's, we're going to skip this just for time. This was just an unbelievable thing that happened during the case. But these were the 3 things that I wanted you, and you can talk about any of these things, but I'm particularly interested in your response to the last one. Number 1 is, what is your initial reaction to this case? What factors allowed for a miscarriage of justice? And I'm just curious, should anyone in the prosecution or the forensics team be held personally accountable for the miscarriage of justice? Why or why not? Why or why not? So what I'm going to do now is I'm going to break you out into the rooms. So again, should anyone be held accountable, and what is your personal reaction? We'll focus on those 2. And I'm only going to give you like 7 minutes in the breakout room, but I just want to see if it works. Okay, stop share. Breakout room. So here we go. So I'm going to do is I'm going to open the room. You're going to see everyone else. I am then, and I want you to discuss those questions. I am then going to move from room to room. So I'm going to come say hi to each room. I'm going to try to. Okay, so here we go. Ready, set, go. All participants have been done. Okay, now there should be a thing to join a breakout room. So go ahead and say yes. You should join the breakout room. Franklin, you need to join. Derek, you need to join. And Peter, you need to join. I mean, Franklin, yeah, good. And okay, great. Yeah, okay. Now. Um, join. There we go.
Yeah. That's so silly.
Hi, I'm here, just in case you didn't know. Okay.
Hello.
Dr.
Just
Weinstein.
keep talking.
Hi.
Or you have any, yeah, just keep.
Okay. So there was a part I remember where they said that the evidence, the handprint, it was actually caused by the soil or like the red dirt, and the spills that they thought were blood spatters, were actually like juice or milk. Like that shocked me. Like, how could you confuse like blood with like things like milk or juice or like
Yeah.
dirt? Like, I was just like, whoa, this is.
No, 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 no. Wait, what are you doing here? Derek, you never found your room. Derek? Derek? What's he doing? What is he doing? How do I send the message?
Hi, Dr.
Hi.
Hong.
Hi.
We were
I don't.
just talking about all the problems
Right.
in the finding of evidence and how it was all flimsy and
Dr. Hornstein, do you think she did it, or do you think the dingo did it?
no.
Really? Really? You have any doubts? Really? Max, you think there's a possibility that she did it?
No. I'm just asking because, I mean, even though it was clear that the dingo did it by the end, there's still obviously a lot of people in Australia who think that she still did it, probably even to this day. So
Right. No. So,
Yeah.
so before I answer, well, obviously you know my answer. What do you think? What is
Are
your
you
honest
to
asking
the
just
group or
Max?
to me?
Sorry, Max.
Oh, I definitely think that the dingo did it because just the case that they presented in their inquiry afterwards was clear that all the evidence that they found to convict Wendy Chamberlain was very false and flimsy. And as I was saying to the other people in the group, it was definitely tailored um, to satisfy the sentiment of the Australian public, who obviously all thought that she did it.
Mhm.
But then when they found like the blood, the infant blood in the car was just spoiled milk, or there's sound dampening material, and then there was, I think, a sand print instead of a bloody stain, and then I think that the forensics team was probably not thorough because the other piece of the baby's clothing was found 200 meters away from where they found
Yeah.
other clothing, and
Yeah,
then
I was.
yeah.
Yeah, that was one of the things that upset me the most. I think was that the investigation teams were definitely at fault there, because if they had really combed the area as meticulously as they should have, especially after the mother mentioned another piece of clothing,
Mhm.
instead of just assuming she was lying, which they kind of did, they assumed since she sounded so cold and emotionless that she
Mhm.
must be lying about the jacket.
Mhm.
It doesn't seem like they even tried to look for it,
Mhm.
which in itself is a huge problem, because if they had found it, the case would have gone a completely different direction, because they would know that she was at least telling the truth about the jacket. But it took them 3 years to find that crucial piece of evidence, which is just insane to me.
Yeah, I agree. Okay, anyway, that's all everything's good. I'm gonna keep going. I want to see the other groups.
Bye.
Talk to you all.
Bye.
Bye.
Um,
Okay, Derek, I'm gonna. How do I go from one breakout room to the next? Are you guys done talking?
Yeah.
Are you all pretty much decided on everything already? You talked it all through?
Yeah.
Yeah.
Okay. Okay, thanks. See you later. One more. Okay, I'll do one more and then we'll go. Oh, God. Um, it's quiet here. Does that mean you guys are all done? You guys are finished?
Yes.
Okay. Okay, I'm gonna call everyone back. There'll be a minute delay and then you'll be called back. Thanks. So how did for those of you who are here, how did the breakout room work? Was it effective? You were able to talk to each other okay? Were there any technical issues?
It was fine.
It was fine?
Yeah, it worked.
It worked? Okay, great. Okay, we're gonna be all back
Yep.
here. Okay, so we all should be back now. Does anyone have any comments about the breakout room itself? Were there any technical, I didn't make it to all the rooms in time, but were there any technical difficulties with any of the rooms that I should know about? Just so, because when we talk about, we're gonna be talking about a rape case tomorrow, and I wanted to make, because the chat room is much more important. I want to make sure you guys can do that. So were there any problems with that? Okay, great. So why don't we talk for a second about it? Just anyone who wants to share, to answer any of those questions, who just wants to share about what they heard in their group or want to share what they think themselves. It brought up, in one of the groups I was talking to, it brought up a lot of feelings. But anyone want to share?
Yeah,
Sure.
I wanted to say that in Australia the juror is very easily biased and influenced by outside information. I'm pretty sure in like important cases in America, they keep them isolated from the outside world so that they can't get any bias to change their
That's right.
decision.
So I don't know whether they have sequestration. Do they not? Do you know for sure that under some cases they don't have? Yeah, but that's right. We
I
have
don't know.
sequestration. Yeah, it's true. And sometimes it gets really crazy where in the O.J. Simpson trial they were sequestered for like, I don't know, several months. They could have conjugal visits like on weekends. So their partner could come and visit them for the weekend. But other than that, they were pretty much alone with the other jury pool. Yeah, and it's a good point, and it does seem to help. It theoretically should help. Yeah. What about the last question that I asked, which was, is someone who makes such incredible errors, should they ever? Oh, Cece, you wanted to say something? Go ahead.
Yeah. It's sort of, I think you were asking if someone should be held accountable, which sort of ties to what I was going to say anyway. That I was saying in the smaller groups that one of the things that upset me most was that they found the jacket that the mother had mentioned so close to the scene 3 full years after she and her husband had been imprisoned.
Right.
And I do think that if they had been, if the investigative teams had been as meticulous and in searching and combing the area as they should have been, especially after the mother mentioned the jacket, they should have been more aware and like actively tried to find it to confirm.
Right.
At least the head of the team should be held accountable, because just that fact, and the fact that all the blood stains they found were not blood stains at all. If not the whole team, then at least the heads or the leaders of those teams should be held accountable for not having instructed a more meticulous search.
And when you say accountable, what do you mean by accountable? Do you mean they should be fired, or do you mean they should be brought up on charges, or?
I don't know about charges. I don't know if it was necessarily a criminal act. I think it was just an oversight, but I think there definitely should be. It should be how do I phrase this?
A fine?
Maybe more. Maybe they could lose their job, but I think there should be more of an investigation into like the reasons behind their oversight and like where it came from. If it was them really being terrible at their job, then maybe they should be fired.
Right.
But I think there would still need to be a sort of a look into the whole situation and like why they were so mistaken.
Okay. Does anyone have any comments on that?
I just wanted to say, in terms of like Lindy, just one part of the video that really stood out to me was the part where she was like talking about how dingoes kill their prey and like how they like strip the skin off, and like it was like a week after her baby got taken, which is kind of where I understood where like the media and the sensationalization was coming from, because the fact that she could talk about, I don't know, like such a horrible thing and like really put detail to it, like kind of put me off, and I was like, whoa, that's really disturbing. The fact
That's
that
cold.
like Yeah, like you could so easily just apply it to like that happening to a baby, like a 9-week-old child, and it's just like so disturbing in my mind. So that kind of like I don't think she did it now, obviously, but like I could see where all of like Australia was coming from when they like saw that interview.
Okay, good. Gracie, did you want to say something?
Um, yeah, I think about the way that they should respond to the people on the forensics team. I think that they should probably, like, at least, like Stacey said, the leader of the team should be fired just because they said there was blood, but they didn't test to see if it was really blood. I think they like denied things that the coroner said that it was a dingo attack. They didn't find the jacket until 3 years later. Like, there's like this level of incompetence that's so obvious.
Yeah.
And then if someone like testified as like an expert witness, I think that could potentially be some kind of legal consequences because like they're not really telling the truth in a trial. Like,
Right.
yeah.
Yeah, Samantha, did you want to say something?
Yeah, I think that the issue was that the people responsible for doing the forensic tests were also the people who are responsible for making the arrests, because it said that the police department are the one who are like testing everything, and I think that's an issue because not only do they not probably have enough training, but they're also pressured to make an arrest, so they may like fake evidence or rush it and not do the test correctly in order to get the outcome they want.
Yeah, yeah, no, I mean, you know, John Oliver talked about this. Was it John Oliver or someone else about the separation of the forensic science team from the yeah, no, it was John Oliver. The separation of the forensics team from the prosecution, and they should be completely neutral, and they're not at all at this point, unless the defense calls its own forensic team, right? But the prosecution forensic team has so much more resources, right? You'd have to be able to pay for your own forensic team. So then again, money is buying something. And O.J. Simpson did that. O.J. Simpson, because he had so much money, you know, he was able to buy all these forensic people to discredit the prosecution's forensic people. But that seems to be the only time you get some sort of balance in the situation. Okay, yeah. I also think we should also think it's easy to judge Australians, but we wonder whether we ourselves could be capable of being of sort of that crowd mentality and be able to, I mean, to think if in your own life, have you ever done something like that, but on a smaller scale, where you went along with the group and were so convinced and you ended up being really, really wrong, but your heart was in it and you felt for sure you had to be right. And I wonder if anyone, I want to end with that. Has that ever happened to anyone where they just got it all wrong because their emotions got the better of them, more in sort of this sort of group situation? Yeah. Okay. Just thought I'd throw it out there. I thought maybe someone raising their hand? It doesn't say it. If anyone's raising their hand, could you speak up? You'll have the last word. No? Okay, I was thinking, do you guys really need an extra day to work on your, I mean, I gave you a fair amount of time to work on your thing. My feeling is we should just keep going with the curriculum. Unless there are how many people would really like to take one more day to work on with their group? I could set you up in break rooms tomorrow, for example, in your groups, and you could just work in your groups in breakout rooms.
Please.
Say yes if you would like that. Okay. Okay. So we're gonna start looking at a rape case, but we'll do that on Monday. We'll start talking about semen. Tomorrow, Monday will be about semen, but tomorrow we will talk about. I will break you out into breakout rooms, and you will do. You'll work on your project. So bring whatever you need to work on the project, and I'll just visit you in your rooms periodically throughout the day. And that's it. So otherwise, I will. Don't forget to sign in the attendance thing if you haven't already, and I'll see you tomorrow. Okay. Bye, everybody.
Bye.
Peace.
Bye.
Oh,